Hello dear student, how are you? I hope you all are fine. Welcome to my class. Today I want to discuss about polymerase chain reaction. What is polymerase chain reaction? Polymerase chain reaction is a molecular biological technique that create or multiply a million copies of DNA. Why it is called polymerase chain reaction? Because the only enzyme used in this technique is DNA polymerase. Basic components of PCR. The basic components of PCR are DNA template, that means the DNA sample, DNA polymerase, it's an enzyme, two primase that determine the starting point of DNA elongation, then nucleotide act as a building block for polynucleotide chain synthesis, then buffer solution, which maintain a suitable pH environment for DNA polymerase enzyme, then bivalent cation. Bivalent cation act as a cofactor for DNA polymerase. PCR is a thermal cycle. The principle of PCR mainly involves three steps. Those are denaturation, annealing, and extension. Those three steps depend on different temperature. In denaturation steps, the temperature is 90 to 95. In this temperature, double-strand DNA convert into single-strand DNA by breakdown the hydrogen bond. In annealing steps, the temperature is 55 to 65 degrees centigrade, and in this temperature, DNA primer bind to the 3' end of single-strand DNA and start the elongation. In extension steps, the temperature is 70 to 75, and in this temperature, DNA polymerase start working and they add one by one nucleotide to complete this synthesis of DNA. After completing these three steps, we can get two DNA molecules from one DNA. By repeating this cycle, we can get multiple copies of DNA from one single DNA. This multiple copies of DNA is the PCR product that called the applicon. Next, the DNA sequencing. DNA sequencing is a method that can be used to sequence DNA molecule. DNA sequencing is very important for agarose gel electrophoresis. Agarose gel electrophoresis is a technique that we can get different band depending upon the different DNA size after completing the agarose gel electrophoresis. For DNA sequencing, we can use two methods. One is Sanger's method and another is Maxam-Gilbert method. Sanger method is also called dideoxy method because dideoxy is used here to break down the polynucleotide chain. Maxam-Gilbert method is also called enzymatic or chemical method because chemical or enzyme used here for DNA sequencing. Both of these methods based on the generation of DNA fragment of different length which start at a fixed point and terminate at a specific nucleotide. Next, the application of PCR. PCR is very important technique in molecular biology. The important application of PCR is genetic fingerprinting. Genetic fingerprinting is important in forensic test. For example, from a crime scene, we can get two types of sample DNA. One is victim and another is unknown sample. But those samples contain very teeny amount of DNA. But for the analysis, we have to amplify those teeny amount of DNA into multiple copies. And in that case, we can use the PCR for amplification. At the same time, we can get or collect different suspect sample. After performing the agarose electrophoresis, we can find these types of band. By comparing those bands, we can easily conclude that suspect 2 is the main criminal of this crime scene because crime scene blood sample DNA band is completely matched with the suspect 2. For that reason, suspect 2 is our target culprit or criminal. Next, the paternity test. Paternity test is useful for comparing the baby's DNA with its parents' DNA, and it is also important for sibling identification. Next, the viral disease detection. You know that RT-PCR is used for coronavirus test diagnosis. What is RT-PCR? We will discuss in our next slide. Next, cloning gene. We know that a gene is used for gene cloning. In gene cloning, we can amplify our target gene into multiple copies by use PCR. And those multiple copies of DNA is very convenient for us to complete the gene cloning cycle. Next, the practical modification of PCR. Here we discuss about two types of practical modification. One is asymmetric PCR and another is RT-PCR. What is asymmetric PCR? Asymmetric PCR is used to amplify one strand of the DNA than the other. We know that DNA is double strand, and in normal PCR cycle, both of these strands can be amplified. But if we want to amplify one single strand of this DNA, then it's called the asymmetric PCR. Next, RT-PCR. RT-PCR means reverse transcription polymerase chain reaction. Reverse transcription. Reverse transcription is a process where mRNA is converted into complementary DNA or cDNA. And in these steps, an enzyme is used known as reverse transcriptase. For this reason, it is called the RT-PCR. And in these types of PCR, the starting material is RNA, and the aim of this PCR is to amplify the particular amount of DNA. The particular amount of RNA. Okay, I hope you all are understand. Thank you. We will see you in next class.